Okay, good evening. If we could have your attention, please. If everyone could be seated. Good evening, everyone. My name's Professor Jim Ogloff. I'm the Dean of the School of Health Sciences at Swinburne, and I'm absolutely delighted to welcome you here tonight for the 11th annual Barbara Dicker Oration. It's hard to imagine that 11 years have already passed with the oration. What I'd like to do, of course, which is our tradition, is begin by an acknowledgement of country. Now, it's particularly special because we also have, in addition to the people who are here, we have about 250 people online from various different areas. So I would like to take the opportunity to respectfully acknowledge the traditional owners of the land on which we gather in Hawthorne tonight, and pay my respects to the Aboriginal community elders, past, present, and emerging, who've resided in this area and have been an integral part of the history of the region. For the online guests who are joining from different areas, if they would like to, they can enter an acknowledgement, just the area that they're in, to acknowledge their local people. And in particular, I'd like to welcome and acknowledge Aboriginal and Torres Strait Islander people who are present physically or online. When we think about health, we certainly do think about the priority around Aboriginal health in both our university but in the broader society. So I want to just highlight a few housekeeping matters which will be of no interest to people online, but the people in the room, we have toilets which are out that door. There are toilets just around the corner. There are 2 exits if there's any emergency, and there is a bar if there's other kind of emergency here. And more food and drink downstairs. And we're going to try as much as we can to keep to time tonight, particularly for people who are online. And as people know, we'll have the oration online and live, and then we'll have a question and answer session following that, and an opportunity to mingle and, in particular, talk to some of the researchers here who are presenting their research, which we'll be able to look at. So we have a wonderful evening ahead. And to begin, it's my pleasure to invite our Vice Chancellor and President, Professor Pascal Kester, to provide a formal welcome and say a few words before this evening's oration.
Thank you, Jim, very much. I would like to warmly welcome on our campus and in this new space that we have devised over the last couple of years, members of the Dicker family. Including, of course, Dr. Ian Dicker. I believe that Tony Dicker is on his way and will be with us shortly, and other members of the Dicker family. And of course, our keynote speaker, Professor Michael Berk. We have 2 visiting scholars from Norway, and I'm going to butcher their name with the greatest of enthusiasm. We have Professor Hedda Beate Soyennansson. Not sure if I get a score for that. And Professor Kristen Nigo-Shoza. And I apologize for not knowing my Northern European language as much as I should. And members of the Swinburne Council, we're most blessed to have with us a number of members of our council, and it's really important because our council is such an important element of both our strategy and of the activity that we undertake. And they are the invisible captains, and we really treasure their attendance at events such as this. And of course, a warm welcome, somewhat wet. It was warm this morning, it's wet now, to everyone here in the room as well as online. This is just such an affirmation of how much better things are that we are in the same room and that we are also welcoming people who are not in this room. I think the first Barbara Dicker oration that I attended was purely online, and I think we can all attest to the fact that it is so much better when it is in 3D. We are very pleased to be able to hold this event in a hybrid way, but we're particularly pleased that we are holding it in this new space, which some of you may discover, and you may not have been in this space before. It's an important place of gathering. It is adjacent to our campus. For many people who have been students at Swinburne University in days past, it is associated with fond memory and with fuzzy memory, depending on who you are. But the reality is that this first floor is, for us, an investment in the capacity to make Hawthorne home, to people who have graduated in the past. We want this space to be a space where, just like tonight, people with common interest, with common passion, with common enthusiasm for what it is that the university does can actually get together and feel that they belong to the family. This occasion today is an important marker in the Swinburne calendar. It's an opportunity to facilitate meaningful conversations about mental health and brain sciences, as well as highlight some research achievements and advancements by our people. It is also a very memorable way to honor the memory of the late Barbara Dicker and her legacy and the inspiring work that her family carries today through the Barbara Dicker Brain Sciences Foundation. The partnership and friendship between the Foundation and Swinburne began more than a decade ago. And over the past 11 years, we have created a strong partnership that spans 3 generations of the Dicker family, many of whom are joining us tonight. The support of the Dicker family enables Swinburne to conduct important research that is tangibly improving the lives of people affected by neurological and psychological disorders. It also includes generous investment in our early career researchers, and this is quite a unique trait, a unique characteristic of that generous donation, is that it assists researchers at the time when they need it most, in the early stage of their careers, where nobody quite believes in them yet, and where an investment in their time now will make such a difference to their trajectory and to the impact that they will have on their domain further on. Many of the 2022 Barbara Dicker Brain Sciences Foundation grant recipients are in the early stage of their research. Some of them are here tonight. And it is this fundamental investment in the future that we so treasure and respect from the Dicker family and the Dicker Foundation. So thank you for your support. Thanks to you, Swinburne has now a great depth of talent and expertise in these very important areas. Now, if we needed demonstration of the importance of mental health and brain sciences, I think the last couple of years certainly gave us much to think about. The plight of people affected with depression and with brain and health, mental health issues has become perhaps more publicly acknowledged, but it is important work, and universities play a big part in trying to solve it. In terms of statistics, and they are sobering, according to the latest national study of mental health and well-being, more than 2 in 5 Australians, that's 8.6 million people, have experienced a mental disorder during their lifetime. In the last year alone, more than 3.4 million Australians sought help for their mental health. This is an issue that touches all of us, either directly or indirectly. So we're particularly thankful for the generous pledge that was made in 2020 by the Foundation for a further 5 years of funding for Swinburne to enable absolutely integral research and development to continue. I am so pleased that the partnership between Swinburne and the Foundation is now so strong, a strength which I believe comes from the alignments of our ambitions. Last year, Swinburne launched a new strategy, Horizon 2025, which is centered on a renewed vision of people and technology working together to build a better world. This vision captures our belief that technology can bring about social and cultural good and be used to solve some of the world's greatest challenges. At Swinburne, we are committed to use technology to advance human health and bring about important breakthroughs to deliver more for those that need care today. We can do this in large part through our partnership with the Barbara Dicker Brain Sciences Foundation. And in the last few years, some of the important technology-led innovations the Foundation has brought about include using mobile apps to improve cognitive stimulation for people with dementia, which we know will be a diagnostic that will affect more than 1 million people in Australia by 2058. A free telehealth service to provide ongoing emotional support to people in aged care residences in response to the COVID-19 pandemic. And a project to monitor activity as an early warning technology for bipolar disorder. Tonight, we will have the opportunity to mingle with some of our researchers and see the outcomes of their work across diverse areas, such as bipolar disorder, depression, sleep, and youth mental health. Outcomes made possible by the investment of the Barbara Dicker Brain Sciences Foundation. Now, we are most fortunate to have internationally recognized leader in mental health, Professor Michael Berk, join us to deliver the Barbara Dicker Oration. Professor Berk, I look forward to hearing more about your work in developing novel therapies for mental health disorders and how these innovations are changing the delivery of healthcare and outcomes for people experiencing the most burdensome of disorders. To close, I would like to thank the Dicker family again for their generosity. We look forward to the wonderful advancements that will be achieved in the second decade of partnership. I also want to thank a wonderful Swinburne philanthropic community. This year, we gratefully accepted an anonymous $1 million gift from one of our very generous alumni, most of which was directed at the foundation. This essential research cannot be done without the incredible support of our generous donors. I encourage everyone joining us this evening to consider how you may also wish to invest in Swinburne's mission of people and technology working together to build a better world. Thank you, and I hope you enjoy this evening event.
Thank you very much, Vice Chancellor. As we've heard and we know, we're really here to honor the memory of the late Barbara Dicker, and we do that partly by the annual oration, and similarly to acknowledge and thank the Dicker family for their generosity, and also the anonymous donor now, who we've heard has contributed significantly to the foundation. So it's my absolute pleasure tonight to be able to introduce the speaker. It's a rare privilege. We often use laudatory language when we talk about colleagues, but Professor Michael Berk really stands head and shoulders above anyone in our field, not only Australia, but internationally as well. So Michael Berk is currently an NHMRC Senior Principal Research Fellow and is the Alfred Deakin Chair in Psychiatry at Deakin University and Barwon Health, where he heads the Institute for Innovation and Innovation in Mental and Physical Health and Clinical Translation, called IMPACT. He's a world authority in psychiatry research, and in 2019 received Victoria's highest honor, the Victoria Prize for Science and Innovation. His findings have influenced treatment strategies literally around the globe. He's an honorary professorial research fellow in Department of Psychiatry at the Florey Institute for Neuroscience and Mental Health, and at Origin Youth Mental Health at Melbourne University, as well as a School of Public Health and Preventative Medicine at Monash University. Michael's recognized as Australia's most highly cited neuroscientist. Professor Berk has authored more than 1,300 papers and is listed as a highly cited scholar in the field. He's been awarded more than $144 million in grant funding, and his major interests, as we'll hear about tonight, are in the discovery and implementation of novel therapies and risk factors and prevention of psychiatric disorders. So it's my pleasure to welcome Professor Michael Berk.
Many thanks for that generous introduction, and many thanks for the honor to be able to talk to you about the work that we do. It's a particular pleasure to be amongst friends and colleagues. I've had the pleasure and privilege of working with many Swinburne colleagues over the years. Greg Murray, who does transformative work in digital interventions for bipolar disorder. Susan, who's my right hand in the Magnet National Clinical Trial Network. And it's also fabulous to see some of my own PhD students, like Amy Haley, who are achieving stunning success. So it's fabulous to be here, and particularly fabulous that I'm able to continue to work with, collaborate with, and partner with Swinburne University in my journey of discovery. So we're interested in developing novel therapies for people with mental health disorders. And we have tried to develop a stem cell to clinical translation bench to bedside platform for the development of novel therapies. Why, you might ask? Well, this is data from the latest census. And as you can see, at the top of the pile is mental health disorders. So mental health disorders have now overtaken everything else to become the leading burden of chronic disease in Australia. That's a problem. What makes it worse is that we don't have good enough treatments, and the pipeline for the development of new treatments is declining, despite what everybody thought would happen. You can see this is data from a biotech company, and every single dot represents a study. So anxiety disorders, you can see in 2000 there are a few, but since 2010 and 2020, there's almost no new development in anxiety disorders. In major depressive disorders, the pipeline is shrinking and there's really nothing new. In bipolar disorder, you can see there's almost nothing happening. This is a major crisis for our field. So I've been working for, it's at least since last week, Tuesday, 3 decades to try and develop novel therapies. So how do you do this? What's the pathway? So in the idealized, rarefied world of academia, what you want to do is find the cause. There's a protein, there's a gene, develop a drug, ping. Except we don't have that for any psychiatric disorder. There's no singular target for anything. So people have hunted, they have not succeeded. So that path 1, ideal, doesn't exist. Path 2, and we're not going to show you this work today, but we've done a lot of work in this area. So you target systems or networks, oxidative stress, inflammation. We think we've had some success in this area, but time does not permit me to share this with you. Sadly, path 3 is where almost everything's come from, and that's interesting. So it's just serendipitous discoveries. People observe weird and wonderful things in clinical practice, and then they follow them up. That's how we got all our treatments. All our antidepressants and antipsychotics synthesizes antihistamines, and we found that they calmed psychotic people, and they chirped up unhappy people. It was a complete accident. And then you say, what is this doing? And you make a slightly better version of the same. Lithium was an accident. But we think there's a better pathway, which is, oh no, I have now. Hang on. We think there's a better way. And the better way, we think, is repurposing based on reverse engineering. And I'm going to just explain to you what we do. So this is the development of validated models for working out agnostically what other treatments do and see, can we come up with treatments that do the same stuff? So we think repurposing is a much more pragmatic way to go about novel therapy development, because we know in the NIH system, in the USA, 30% of new drugs approved by the FDA are repurposed. They're not new chemicals. There's a lot of success in areas from cancer to immunology to infectious diseases. It's much cheaper and it's much quicker because you know the drugs, right? These are known agents, but we find new uses for old dogs. So let me just tell you in summary what our model is, and then I'll give you 3 examples of how we've been able to take this model and develop new therapies. So we start with stem cells. What we do is we take stem cells from patients, take white cells, turn them into stem cells, and grow mini brains in test tubes. Then we tickle these cells with medications, and we see what do they do to proteins, gene expression, things like that. We then get what we call a gene expression signature or a proteomic signature or whatever signature, and then we can say what other drugs do the same thing. We then go to animal models, so the animal models of mania, animal models of depression, whatever, and we say, does this show the same signal? Does this replicate? Does this confirm our preclinical signal? We then, for some drugs, we can go to epidemiology, so we can look at big population samples and say, in the whole of Sweden, if you take this drug, is your risk of developing disorder X or Y, more or less? And then we can go into clinical trials. And clinical trials is what I'm doing together with Susan. So Susan and I lead Magnet, which is Australia's National Mental Health Clinical Trials Network. And this is where we really kick the tires on any of these bright ideas. So I'm going to start with angiotensin. Any of you who went to medical school would know angiotensin has absolutely nothing to do with psychiatry. This is in the renal textbook. It's about blood pressure, and it's got absolutely nothing. If you're from psychiatry, you do not have to read this chapter. Well, maybe you do. So how do we stumble onto this? Well, it all started with a paper produced by colleagues that I was not working with. These are an independent group at Deakin University. So they took Wellcome Trust genetic data. So they looked at all the genes that are associated with a certain disorder, in this case bipolar disorder. They looked at the matrix of genes that are associated with bipolar disorder, and then they can look at bioinformatics to see are there any medications that target these genes. So this is purely an in silico computer simulation. And then you can see, does anything match? And they come up with 2 hits. The one was, imipramine. Now, imipramine, those of you who know medicine, is an old antidepressant. It's a warhorse. We know it works. Statistically, you can think of this as a positive control. And then we come up with valsartan. That's an angiotensin drug. It's a blood pressure medicine. Surely that's a spurious, irrelevant lead. Well, we ignored it. Nobody paid any attention to this at all. Went into the shelf. A couple of years later, I went to listen to a lecture by the person who's now my co-director of Impact, Ken Walder. And I keep on saying, this is the best lecture I heard in the decade. So Ken's doing this work in diabetes. He's a diabetes researcher. He's trying to develop treatments for diabetes. He was telling me what he does in diabetes. So what he does is he takes stem cells from diabetics and he tickles them and he sees what genes wake up. And then he goes to a compound library screen and he sees what other drugs in a compound library of 2,500 drugs do the same as all the anti-diabetic drugs, and he comes up with a glaucoma drug. Now, glaucoma is pressure in the eye. What's this got to do with diabetes? Nothing, of course. Then he goes to animal models of diabetes, and methazolamide lowers glucose, which is what it's supposed to do. It's an anti-diabetic drug. He then does a clinical trial in humans and shows that methazolamide is an effective anti-diabetic drug. Now, I just fell off my chair when I heard this work because, in a way, diabetes is just like psychiatric disorders. It's a complex, polygenic, environmentally mediated disorder. So same risk factors, very similar gene-environment interactions, and I thought this is brilliant. We have to do this with psychiatry, and for the last 10 years, we have. So let me tell you what we do. So please don't be distressed by these pictures. They're not as complicated as they look. So we take people with bipolar disorder, we take bloods, we grow stem cells, and that's what they look like under the microscope. We differentiate them into cortical networks. So this is a mixture of neurons and astrocytes. We think that the choice of cell model is terribly important. So we initially started by going to a commercial lab and buying off-the-shelf neurons. You can do this, right? You can buy a cell line. We now think that's not such a fantastic idea. Not that we didn't get useful leads, we did, but we think that real cells from real people is much more useful. So when we use this model, what do we come up with? Well, I don't want to take you through this whole list, but I'm just going to take you for the three in the bottom. So cenarizine. Now, this is a calcium channel blocker. Why is this relevant to bipolar disorder? Well, I saw cenarizine and I thought, whoopee-doo, because little on me, what did I do my PhD on? Calcium. Intracellular calcium. It's elevated in bipolar disorder. It's a very important signaling molecule. So if our first drug is a calcium blocker, that's interesting. Our second drug is flupentixol. It's an antipsychotic. And we know it's an effective anti-manic drug. So it's another, what I would call a positive control. The third drug we come up with is racemine, another angiotensin drug. So we now have two atheoretical drug screens, and we come up with an angiotensin drug. We also have valdecoxib, and there's a lot of work. It's an anti-inflammatory, a lot of work showing that these drugs might have antidepressant effects. And then there's a whole lot of cancer drugs, and we have no idea what they're doing there. While we were watching, there's a group in the US who's doing very similar stuff. So they were looking at major depression. We were looking at bipolar disorder. But they do very similar things. So they looked at the genes involved in treatment-resistant depression. They took the genes in samples with major depression. They looked at genes targeted by drugs, and they looked at the drugs that tickle those genes. And what do they come up with? I should have put it in color. Angiotensin drugs, telmisartan. They also come up with a whole lot of other candidates that are currently under very active current investigation. So they come up with cannabinoids, and there's people in this audience who are studying cannabinoids. They come up with ketamine. Well, this is the hottest new thing on the block. They come up with pramipexole, and we already know that this is an effective antidepressant agent. It's an old Parkinson's drug. So a whole lot of drugs come up here, which basically represents what's currently under investigation, plus they come up with angiotensin antagonists. So we were very excited. This again confirms this. So three completely different screening paradigms come up with the same drug. So now it's time to look at animal models. So we worked with a group in Texas. This is a group in Houston. And there's an animal model of mania. You give animals amphetamines and they go manic. They run around and they don't stop and they don't sleep. But if you give them, and you can see that amphetamines massively increases activity. If you give them lithium or an angiotensin drug, candesartan, you block this. So the angiotensin drug, candesartan, blocks mania in this animal model. There's also depression models. There's a couple of different depression models, and I'll just briefly talk you through them. Not that you're terribly interested in animal models of depression, but I think it's just to understand what we do. So one of these is a test called the sucrose preference test. So in theory, depressed rats don't drink much sugar water, whereas happy rats do. So if you stress a rat, it stops drinking sugar. And if you give it candesartan, it starts drinking sugar again. And if you give it any known antidepressant, they start drinking sugar water again. So in this model, comes up ping. It's got antidepressant properties. Now we go to humans. Now, in epidemiology, one of the best class of drugs to study is antihypertensives. Because in clinical use, you can pick beta blockers, angiotensin drugs, calcium channel drugs, and it's just random which one you choose. So this is data from the whole population of Norway. We have very good epidemiological links with colleagues in Norway. And we look at beta blockers, and they make you more depressed. And if you take a calcium channel drug, it doesn't do anything. But if you take an angiotensin converting enzyme inhibitor or an ACE inhibitor, your risk of depression is just about halved, which is fantastic. I mean, that's really exciting. So that was the earlier study. I don't want to bore you to death, but we've now replicated this 10 times in 10 different epidemiological studies. This is from our Geelong study. So we have a big epidemiological study in Geelong called the Geelong Osteoporosis Study. Ignore the name osteoporosis. That's one of the things we do, but we do other stuff. And you can see this dramatic, I've never seen a curve like this, but in this curve on the red, you can see people who are taking the drug and they simply do not become depressed. It's quite remarkable. If you don't take it, there's a decay curve to people becoming depressed. There's a lot of literature in the cardiovascular literature where they use it for heart failure and they use it for hypertension. And the cardiologists measure things like quality of life. Now, quality of life as a psychometric construct looks very much like depression. And what you can see here is a significant improvement in mental well-being, positive well-being, overall quality of life, suggesting that in randomized placebo-controlled trials, angiotensin drugs improve quality of life, which is, as I said, very similar psychometrically to depression. So add this all up together. This was enough to convince the NHMRC to fund 2 clinical trials, one in unipolar depression and one in bipolar depression. And these studies are running. We have 3 study sites in Melbourne, one at Eastern Health with Judy Hope, one at Melbourne Clinic, one at Albert Road Clinic. So the study is currently underway, and we're studying both unipolar depression and bipolar depression. So that's my first example of this stem cell to bedside translational pathway. Again, if it's effective, it costs $5 a month. Its side effect profile is better than any antidepressant. And if this works, the path to translation should be relatively smooth. Chapter 2, metformin. Now, those of you who went to medical school might know that metformin is a diabetes drug. And this is for sugar. Again, it has no place whatsoever in a psychiatric lecture or in the psychiatric curriculum, and you surely need to know nothing whatsoever about metformin. Maybe not. So our stem cell model identifies metformin as a drug that comes up as having very similar properties to known antidepressants and bipolar medications. And I'll just take you through our journey again. Apologies for the slide, it's a little bit messy, but I will just summarize it so you don't have to pay too much attention to it. This slide, we have information on 3 different tests. Forced swim on the left, which is a test of depression. Sucrose preference, which is another test of depression. And pre-pulse inhibition, which is a test of preclinical screening for schizophrenia. And the bottom line is metformin comes up positive in all of these assays as having antidepressant and potentially antipsychotic effects, which is really kind of interesting. One of the earliest studies was done by a guy called Mark Valquist, who's actually an Australian, but he did this in Taiwan. So he looked at the entire population of Taiwan. Again, Taiwan's one of these places where you can do really good epidemiology because they have fantastic clinical records. And what you can see here, just focus in the top left corner here. If you see, these are the odds ratios of being depressed. In the general population, you can see on the left, it's an odds ratio of 1. In other words, that's even odds. If you're diabetic and you're not taking these drugs, your risk of developing depression is 2.5 times greater. And if you're taking metformin, you're back to normal, right? Your odds are the same. So this was a very interesting epidemiology study, and it had no impact at all. It didn't change practice. Nobody paid much attention to it, but I thought it was a brilliantly designed study. We then went to our own epidemiological cohort. So this is data that was published last month. First author is Lina Williams, epidemiology lead in our group. So here you can see in the red people who were taking metformin, and in the blue, general population people who were not taking metformin. Now, if you think about it, if metformin did nothing, you'd expect it to be worse. And the reason why is that the things that make you diabetic are being overweight, which is a risk factor for depression, being physically inactive, which is a risk factor for depression, eating lots of junk food, which is a risk factor for depression. So the fact that the metformin group is better than general population is quite striking, because it's fighting against this headwind of the risk factors that make you depression, that would make you otherwise depressed. So we thought this was kind of an interesting finding. And about 2 months ago, a really nice paper came out from a lady called Cynthia Kalkin in Canada. So she took a cohort of people with bipolar disorder who were insulin resistant, and she gave them metformin. And what she found was that in people who became insulin sensitive again, because that's what the drug's supposed to do, it takes you from insulin resistant to insulin sensitive. It's an anti-diabetic drug. Their depression scores plummeted. So you saw this dramatic improvement in depression. People given metformin who responded to who became insulin sensitive. It doesn't. The problem with this study is that it doesn't apply to everyone. So we don't know whether this would work in the total population, but the fact that it even works in insulin-resistant people is very interesting, because it was placebo-controlled. So as part of Magnet, this is together with Susan, and ably aided by Ravi and Denny sitting at the back, our fabulous statisticians on the Magnet studies, we're doing a study which we're calling Hearts and Minds. So Hearts and Minds is a study which compares 2 drugs and placebo. So the aim is to compare whether statins, and again, for time pressure, I haven't been able to explain to you why statins might be antidepressants, but I have probably more evidence that statins might be antidepressant than I have for metformin. But just to keep things manageable in time-wise, you'll have to take it as read that there's good evidence that statins have antidepressant properties, including a study that we published a couple of years ago, a randomized controlled trial, which we did at Origin. So we're comparing statins and metformin for the treatments of adults with MDD who are resistant to therapy. And we're trying to see that statins will reduce depressive symptoms, and we're also going to see whether statins and metformin reduce biomarkers that we know are associated with depression risk. So this is a study that is commencing. We should start March. Yeah, we're thinking we'll start in March of next year, once everybody's back from the Christmas holidays and the year has started, and we got our supply and ethics and everything else done. So if that's not crazy enough for you, let me talk about the craziest of the lot, which is mitochondria. Mitochondria have absolutely nothing whatsoever to do with psychiatry. They are muscle things, right? Except they're not. We've been very interested in the mitochondrial hypothesis of bipolar disorder. Now this might come as a bit of a bolt out of the blue, because this is not mainstream thinking at this point in time, although there's a lot of the people who I respect highly in the field have been looking at mitochondria in bipolar disorder, and we stand on their shoulders in understanding where we're coming to. But if you look at the symptoms of bipolar disorder, it's an energy disorder. When you're depressed with so bipolar is highs and lows, right? When you're depressed, you can't get out of bed, you can't think straight, you're exhausted. Just going to the bathroom is an effort. And when you're manic, you can't sit still. Your brain's on fire. You're racing. Your thoughts are going 1,000,000 miles an hour. You can't sit still. You're charging. If you put these people in the scanner, when you're manic, your brain's on fire. It's burning energy. And when you're depressed, lights are out, nobody's home, in terms of neuroimaging findings. So there's pretty good evidence that mitochondrial function is heavily involved in bipolar disorder. So we've developed a model where we think that mitochondria are at the heart of bipolar disorder. Because essentially, to understand bipolar disorder, you have to understand that there's a toggle switch between mania and depression, right? We got a project grant with colleagues at St. Vincent Institute looking at what we think is a candidate toggle switch, but that's too much detail for today. But something is switching you from a state of increased energy generation to a state of decreased energy generation. Oh, what I forgot to mention is that the brain is by far the body's most metabolically active tissue. The body, even though it's 1 kilo, just over a kilo, consumes more than 20% of the body's total energy. It's the most energy demanding tissue of all. In kids who've got a mitochondrial dysfunction, the brain is the most profoundly impacted organ. So what we know is that all the molecular pathways, and I'm not going to explain them to you because that's too boring, but all the molecular pathways that we know are involved in bipolar disorder converge on the mitochondria. Some, if you look at little bars in the middle, are inhibitory, and with little arrows, they're stimulatory. But pathways either converge to stimulate you into mania or flip you down into depression. Exactly what the toggle switch is, we're not sure of, but we are honing down on what we think might be viable candidates. But it's clear that it's a regulator. It's a regulator. It's something that, it's the toggle switch. There's a lot of work going into, and I think more work needs to go into understanding what that toggle switch is. The nice thing about mitochondria is that they're druggable. Right? You can give things to mitochondria to either stimulate mitochondrial energy generation or suppress mitochondrial energy generation. So if you look at this down the bottom, there are a couple of drugs. So you can see, for example, some of the fibrates which you use for cholesterol. You can see coenzyme Q10, lipoic acid, N-acetylcysteine. All of these agents have an effect on mitochondrial function. And we had the thought, well, why don't we come up with a cocktail? We'll take the most available, safe, tolerable ones, put them together in a cocktail, and surely we'll be able to treat bipolar disorder. Well, maybe. So this was our study. We did a randomized control trial of N-acetylcysteine alone, which itself has significant mitochondrial and antioxidant properties, and we compared it to placebo and our mitochondrial cocktail. And the major elements of our mitochondrial cocktail were NAC, carnitine, ubiquinone, or coenzyme Q10, you'd see this on the shelves of your pharmacy, and alpha-lipoic acid, which you'd see in the shelves of your pharmacy as well. So it was a 4-month study with a 1-month follow-up. So what did we find? Not what you'd expect. So this is our results. What you can see here is that at the primary endpoint of the study, week 16, there was no difference between the drugs. It didn't work. What we did see after that is 1 month later, the mitochondrial cocktail was much better. The depression symptoms had improved significantly. Hard to explain why, but that's what we found. What we also saw was a slight increase in manic symptoms. Not significant, but you can clearly see that manic symptoms increased, which I think is proof of principle that the concept is valid. You know, if you can give the cocktail and drive people up, that suggests that it's doing something. But weak signals, but it's kind of hinting you in the right direction, but not quite there. So there's now evidence of a range of mitochondrial cocktails, agents from N-acetylcysteine to melatonin. At this stage, the most potent seems to be N-acetylcysteine, which you can see up at the top. But that's not the only mitochondrial stimulator. Exercise is a pretty good mitochondrial stimulator. And I haven't got time to talk about it, but you should get your patients to exercise if you're a clinician, and you should exercise if you're not. And what we did find in our study was that those people who were physically active and who were on the mitochondrial cocktail on the right-hand side had by far the most robust benefit from this cocktail. So that's a really interesting finding. So the last finding I'm going to show you is from another gene expression signature study of ours, where we identified a very interesting drug, which is called trimetazidine. You would never have heard of it. It's not available in Australia. It's available in Europe. It's a drug for heart failure and angina, and it works by increasing the efficiency of energy generation in the heart. So it's a mitochondrial drug, boosts energy generation in the heart, and it fixes heart failure, it fixes angina. So it does a number of things. It reduces apoptosis or cell death. It enhances energy sufficiency. So we did some preclinical studies, and the first thing we did is we actually showed that trimetazidine increased oxygen consumption. So that proves that it is increasing energy generation in the mitochondria. We were then also able to show that it had beneficial effects in the forced swim test, which is the antidepressant effect, in the sucrose preference test, which is another antidepressant effect, and the elevator plus maze, which is an anti-anxiety effect. So that suggests that this drug might be doing something. There's another animal model that we did, which is in the chronic restraint model. Let me just skip this one for the fact of time. The other beauty of this is it's really well tolerated. So this is a massive study of almost 4,000 people published in The Lancet of trimetazidine. And this is the side effect data. And basically, it's about as clean as a whistle. There's almost no meaningful side effects which separate from placebo, and that's a massive sample size. So it's got promise, and it looks like it's really well tolerated. And we've just got funding to commence a new clinical trial of trimetazidine in bipolar depression. This is a multi-center international trial, which has been done in India, Australia, and Spain. So another really exciting lead. And in 5 years' time, I might even have results for you. So let me finish by summarizing that stem cell models show great promise for the candidate molecule detection. So we think that our stem cell model has given us really exciting leads, and we are actively working on our stem cell model. I'm talking about Ken Walder. He's the brains behind this, not me. And we believe that inflammation, neurogenesis, mitochondrial function, oxidative stress, these provide many targets for therapy development. So there are a lot of exciting drug candidates out there. So they include statins, which I haven't shown you the data, minocycline. Olivia Dean in our team has got some great data on this. Celecoxib, which is not work we do, N-acetylcysteine, metformin, and angiotensin drugs. We think diet and exercise are really important. Additional strategies, again, we don't have time to show you data that we've generated in that domain, but it's promising. And we think that drug repurposing bypasses many of the pitfalls in drug discovery, and I think it might, if we're lucky, contribute to reflowering after the drought. Thank you for your attention, and thank you for the honor of addressing you.
Okay, well, first we want to thank you very much. We'll thank you more formally a bit later, but you still have work to do. So we'll thank you in the first instance for that very stimulating talk. And we do have some time for questions, both from the people present in the room, but also people who are online. So if you do wish to ask a question, we'll circulate the microphone, and also you've got the mic. Mike's got the mic, and you've got the mic, John. So why don't you start with your question?
Thank you very much, Michael. It was a great presentation. Brought back some very bad memories of doing pharmacology and statistics at university. What I am interested in is you mentioned metformin, and you also mentioned depression. Have worked in the past in Indigenous health, and I was wondering whether any of your previous work has had an impact on Indigenous clients, especially given that type 2 diabetes is rampant, and so with some mental health disorders.
That is an incredibly important and prescient question. You are absolutely correct that obesity, the metabolic syndrome, cardiovascular disease is hugely disproportionately present amongst Indigenous populations. So we are, in terms of our trials, we are particularly looking for people with lived experience, and we And in our magnet network, for example, with Susan, we are actively reaching out to get the viewpoints and input of the indigenous population into our studies. And we think that because these agents are generally very safe, they might have significant translational value. Because not only are they might they be useful for psychiatric illnesses, but they're of established value for these medical non-communicable disorders.
Thank you, Professor Berk. I'm the voice of the online questions. And there's a great question that's come in, which is, how much physical activity does it take to make an impact on mood?
Less than you might think.
Excellent.
What's clear is you do not have to do triathlons. You do not have to do ultramarathons. You don't have to do anything heroic. If you do 45 minutes of moderate physical activity 3 times a week, you are doing great. And that depends on who you are, right? So if you are older, taking the dog for a walk for half an hour, for an hour, that's probably all you need to do. If you're younger, you might want to do something more vigorous. But I don't think it matters hugely what exercise you do. It could be yoga, it could be cycling, it could be pumping iron. Everybody has a favorite about what they like to do. But as long as you're doing something. But the key for me clinically is not to overdo it. If you say, I'm going to aim to do a triathlon and I'm going to do 1 a month and I'm going to train 4 hours a day to win, forget about it. It's not sustainable. You'll injure yourself. You won't keep it up. The key is to have goals that are modest and are sustainable and attainable. So taking the dog for a walk 3 times a week, that's attainable. You might want to speed it up, you might want to go for a jog, you might want to go on a push bike, but it's moderate exercise, attainable exercise, reasonable goals. Sustainability is much more important than performance.
Gavin?
Michael, thanks very much for such an exciting and cardiovascular related presentation. The drugs that you were showing, angiotensin blockers, ACE inhibitors, etc., and many people would commonly have and be using those particular drugs. Do you think that the mechanism of those drugs in treating mental health issues is the same mechanism associated with blood pressure reduction?
That's an excellent question. I think the answer is not, because we don't know. I mean, let's be honest. This is an atheoretical paradigm. It's not a target. It's not a single target paradigm. So my own sense is the classical paradigm in psychiatry is you discover something accidentally, and then you go and explore its mechanism of action. So having discovered antidepressants accidentally, you then say, oh, these drugs affect serotonin. Having accidentally discovered antipsychotics, you say, oops, they affect dopamine. And therefore the dopamine theory of psychosis is born. So my own sense is that a lot of this has got to do with the fact that in a Lego block-like manner, agents that have signaling capacities in one tissue would have parallel signaling capacities in other tissues that are not well understood. Angiotensin is probably very important in terms of the stress response system. In terms of statins, I don't think we really know. We started off with an anti-inflammatory hypothesis. We think that they're anti-inflammatory, and that was why we went to them. But I don't know if that's true. One of the things we found, and again, I can't show all this data, but if you look at the pathway that is most disrupted in bipolar disorder, across every element of the pathway, it's cholesterol biosynthesis. Now, we never thought we'd find that, but we did. But so to find a drug like statins that might have effect, well, that's really interesting. And maybe there is something about lipid biosynthesis that is dysregulated at a very core level in these disorders. This is work this is next level work. If we can identify a clear clinical target, we then, in a very iterative, circular way, go back to the lab. So one of the things we're doing, and we're doing this with Susan in the Magnet Network, is we're doing biomarker assays in our things, and then we go back and explore candidate biomarkers to see if they're correlated with drug effect. But my goal is really to come up with something that helps people. Right? I'm interested in, if there's a choice between understanding arcane biochemistry and coming up with something that works but you don't really understand it, I'll do the second. So we start there and then we move on.
Well, we have time, I think, for maybe 1 or 2 more questions, depending on the time. So the online question?
Yes, thank you. Another question, and this has come from Nick. Has there been testing of these novel therapeutics among cases with psychotic features?
Yes, there has. There's one study of telmisartan in people with schizophrenia, and it's a positive trial. So it seems to help particularly negative symptoms in people with people with schizophrenia. Statins are actually quite well studied in schizophrenia, and they also improve negative symptoms in people with schizophrenia. There's now about, I can't remember exactly how many trials, there's about half a dozen, and there's a consistent signal on meta-analysis that statins improves symptoms of schizophrenia. Not dramatically, but it's a 5-10%. So the answer is very much yes.
Thank you. And another question. Interestingly, many people with depression will be on a lot of these medications due to comorbidities, yet their mental health system symptoms are persistent. Have this cohort been studied in terms of understanding why their mental health symptoms are still persistent?
I think that that's a really good question. What troubles us in the mental health space is not that people who have comorbid diabetes or hypertension are treated. It's the fact that if you have comorbid diabetes or high cholesterol and you have, for example, schizophrenia, you're much less likely to receive these drugs than somebody in the general population. So the quality of medical care in people with serious medical illness is actually worse than the general population. And these people are relatively undertreated for significant medical comorbidity compared to the general population. Now that is a service challenge, and people in our group like Alison Jung are working very hard to try and look at how can we improve pathways to better care of medical comorbidity. But the care of medical comorbidity in people with serious mental illness is a substantial unmet need.
Okay, we may conclude the questioning now and then move to we're going to end the live feed very shortly and then move to having the fireside chat without a fire. Sorry, did you have one more? One more. Since they're online, they won't be able to participate later, so we'll make an exception.
Thank you very much, Professor. Within older persons' mental health, how do these medications and therapies vary to individuals who are of younger ages? Is this an area that will be investigated further?
I don't think we have enough evidence that compares people at different ages. Our initial study of, for example, statins was in youth depression. We are involved with studies that are ongoing. Sophia Zungas at Monash is running a fantastic study of statins in the elderly. I would be surprised if we saw a difference, but you never know. I'm always surprised by what we find. You never find what you think you're going to find. But I would imagine it would be the same.
Good. Okay, thank you. I'd like to now invite the Deputy Vice Chancellor Research, Professor Karen Hapgood, to provide a formal thanks.
Well, I'd like to really thank Professor Berk for his really outstanding presentation, walking all of us through, regardless of there are experts in the room and there are interested normal people also in the room and online. And I think there was information in here of all of us learnt something about the creative ways that we can look at new approaches, because the current pharmaceutical discovery and development process has limitations. And there are other options out there that we can look at. And the way it also has the advantage of not only looking at things that have already known and developed, but also that sense of clinical trials you can do in population studies with existing medications that you cannot do in a phase 3 clinical trial when you're in a race to get a new drug to market. So it offers up new and better ways to look at what are the options and ways that we can build that. It's really an important part of research excellence in Australia and also in mental health and with impact across the world. And I'd really like to thank Professor Berk and his and friends who are in the audience for their work and impact on this area, and to also thank the Dicker Foundation for sponsoring and supporting this event. So we've got a small gift with my assistant who's going to deliver by the looks.
It's a small stage. Well, thanks very much.
So that concludes this part of the evening, and thank you very much to everybody who has watched and dialed in online to watch this event, and we hope you enjoy your evening. Thank you very much.